Hi, thanks for inviting me. And I'm going to talk about the use of time series to understand something that we don't usually use time series for. So to constrain the horizontal variability of open ocean ecosystems, particularly related to mesoscale dynamics. The environment that I'm talking about is the North Pacific Subtropical Gyre that has been defined as the largest biome on Earth. And in this MODIS composite image of chlorophyll lake for 15 years, we can see that these regions of relatively low chlorophyll concentration. And the advantage of, one of the advantages of studying this system is in itself the presence of the Hawaii Ocean Time Series. It is one of the most important open ocean time series that lasted for 30 years now with almost monthly, quasi-monthly sampling. This takes place here at Station Aloha, about 100 kilometers north of the island of Oahu in the Hawaiian archipelago. And so this presence of biogeochemical, hydrographic, and ecological measurements allowed us to understand a big deal about the seasonal variability, the interannual variations at this site, and also the vertical variability in particular of this system. But not necessarily anything about the horizontal variability, because from a Eulerian time series perspective, horizontal variability is most of the time thought more to be a hassle rather than something that we target. But here I will argue that we can understand something about this variability using an Eulerian time series combined with satellite observations. So the spatial variability that we have at a site in the open ocean north of Hawaii is in great part driven by the currents in the ocean. And we can imagine that 90%, about 90% of the energy of these currents is in scales between tens and hundreds of kilometers, that is called the mesoscale. Here I'm showing you a plot of sea surface temperature. The advantage of this product is a cloud-free product distributed by NASA. So in this day, we can see that there is a big deal of variability. And even more, if we play the video, here. We can see that this is a period of 3 months. And there's a lot of variability related to these meandering currents, filaments, and mesoscale eddies, so closed circulation feature. But if it's easy to understand this, to observe directly from satellite the variability of the hydrographic field, it's much more complicated to understand what is the biogeochemical variability, in particular far from the surface in this system. And just to try to understand what kind of variability we can expect, this is a very descriptive explanation of some of the phenomena associated with mesoscale dynamics. So I conceptualize a static ocean versus an ocean that has a mesoscale current within it. So in the static ocean, we can expect there not to be currents at the surface, close to the surface, and that the sea surface is flat. And also, if we look at layers associated with a certain density in the ocean interior, we can see that also those layers are flat in the horizontal dimension. While when we have a mesoscale current, conversely, this current is associated with changes in the slope of the sea surface, with a slope in the sea surface, and also an opposite slope in these isopycnal layers in the water interior, so displacement of water layers in the ocean interior. And while there are changes in the order of centimeters in the sea surface, the changes in the depth of these layers is in the order of meters or tens of meters. So this can have important implications. I will go quickly on this one, but empirically this has been proven to cause changes of 2 to 4 meters about in the depth of these isopycnal surfaces associated with each centimeter of change in the sea surface side, both at the Bermuda Ocean Time Series already in 1999 by Siegel et al. And the Hawaii Ocean Time Series in this analysis that I'm presenting here. So it's an important change in the depth of these layers. And the important part of this is also that sea surface height can be easily right now be measured from satellite. And each of you can go and download these maps of global ocean sea surface height. In this case, we are talking about sea level anomaly. That is just the anomaly of this height over a certain time period. And so it's easy to kind of study the effect that these changes have on a certain ecosystem if we do repeated measurements. And to understand what these changes in sea surface height and changes in the depth of water layer, what kind of effects these changes can have on the ecosystem of stationary law, I will start by just saying what are the basic characteristics of this system. These are the median profiles for the whole time series. We have nitrate plus nitrate here, chlorophyll, and one compound, dissolved organic carbon, that is representative of the organic matter in the ocean. But so we have a surface layer that is nutrient depleted, so there's one or few nanomoles per liter concentration of nitrate plus nitrate. Then the concentration starts to increase at a certain depth, around 100-meter depth. So we have this nutrient line, and there is a diffusion, turbulent diffusion of nutrients upward. And the layer that consumes this diffusive flux is associated with a chlorophyll maximum, so where the phytoplankton community lies on top of these nutrients diffusing from below. And this is also a characteristic feature of this system. While organic matter generally decreases with depth in environments like stationary law. So to understand what happens when water layers are displaced, we can start from this kind of observation, that the nutrients are depleted and increase with depth. And already in the 90s, this was conceptualized. I took this from McGillicuddy et al. From 1998. So the idea is that when you bring nutrients up and you expose them to light, the community responds. So there is phytoplankton growth is stimulated by this phenomenon, while there is a very small response where you downwell water. So you put a water that is already depleted in nutrients at lower light intensities. But the surprising part is that it's very hard to observe this phenomenon in terms of blooms. And this has been explained because it is thought that the phytoplankton can respond faster than the rate of nutrient injection, so that at the end we don't see increases in biomass, or it's very hard to see them. But so now I can tell you the questions that I started from. And one is, in an environment like stationary law, do we measure this phenomena of enhanced new production due to nutrient injection in the euphotic zone? But also, more in general, what are the changes in the biogeochemistry and ecology of stationary law that we observe related to changes in sea surface height? And to address this question, I did two kinds of analysis. One is a look at the changes in the biogeochemistry of stationary law related to changes in sea surface height. And the other is actually targeted observations in mesoscale eddies, and in particular in mesoscale dipoles, that are systems made of adjacent eddies of different polarity. So let's start from part one. This is a very, if you want, simple analysis, because I looked at the hot biogeochemical, hydrographic, and ecological observations, and in the first round of analysis, I really looked just at correlations of these observations with changes in sea surface height after downloading the time series of sea level anomaly at stationary law. But really what I'm showing here is something that is visually more easy to understand. And it's the comparison of the top and bottom quartiles of the concentration of stocks or abundance of organisms in condition of high sea surface height that I will represent in red, that are those conditions where sea level anomaly was above 5 centimeters, and low sea level anomaly. So it's this bottom quartile where sea level anomaly was below minus 5 centimeters. So about the first question that we asked, that is, do we observe these events of nutrient injection that stimulates the community? We can look at these and answer this question by looking at the variation in the concentration on isopycnal surfaces that are those that are displaced up and down in the water column. And if we look at them, in this case, I plotted 3 of them. I remind you that the red would be cases of high sea surface height where water layers are depressed, while the blue ones are low sea surface heights where water layers are brought up. So we have inorganic nitrogens in terms of nitrate and nitrite, dissolved inorganic carbon, and oxygen here. So when we bring water up, the anomaly of both the inorganic nitrogen and dissolved inorganic carbon is negative, meaning that there is less in that layer, while the anomaly of oxygen is positive, meaning that there is more. And this is consistent with the fact that you expose these high nutrient waters to light, and you consume the inorganic nitrogen and carbon and produce oxygen. So these are a clear signature of past events of new production. But if then we look at the distribution of chlorophyll and primary production associated with these top and bottom quartiles, we see very little differences, meaning that somehow it seems that we miss the action. So we don't observe, as already proposed in the past, it's hard to observe these events in terms of blooms. But more in general, I was also interested in understanding what are the baseline changes, the general changes in the biogeochemistry and the ecology of stational Loa in different conditions of sea surface height. And I'm here showing some examples. I have no time to go through all of them, but these are something that can be interesting. There are 3 plots here of nitrate plus nitrite, phosphate, and eukaryotic phytoplankton. So we start from the simplest, nitrate plus nitrite. It's consumed very fast in the upper 100 meters, so we don't see changes over there. But we can see that in the deep water column, there are changes in its concentration consistent with the displacement of water layers, so higher concentrations where you have lower sea surface height. And also note that these vertical gradients of concentration increase, so likely the diffusive flux of nutrients increases too. The same is true in part for phosphate, where in the deep waters you have this increase of concentration at decrease in sea surface height. But this is a very enigmatic result in the surface that when you bring these deep nutrient-rich waters up, you instead have a lower stock of phosphate in the upper 100 meters. And this is something that we don't have a mechanistic explanation right now, but we are trying to look into different hypotheses that could explain this. And the third result that I want to show here, it's about the abundance of eukaryotic phytoplankton. It's not very clear in this plot, but I will come back to this later. But you can see that the abundance of these organisms, this is from flow cytometer, the abundance of these organisms is very similar in the 2 cases, apart from at close to the depth of the chlorophyll maximum, where at lower sea surface heights you tend to observe higher abundances. And this seems to be connected with, at least in my mind, with these higher vertical gradients of inorganic nitrogen concentration. But so more in general, to summarize other results that I didn't show, there are depth-specific changes of the biogeochemistry of Aloha with sea surface height. There is a layer above the deep chlorophyll maximum where we see higher stocks of both phosphate and also of dissolved organic nitrogen at higher sea surface height. Then we have the depth of the layer around the deep chlorophyll maximum, whose depth changes and whose depth increases with sea surface height, and where we measure lower abundances of eukaryotic phytoplankton at higher sea surface height. And then there is the layer below the deep chlorophyll maximum that is more boring because it's just what you expect. So inorganic nutrient concentration decreases with sea surface height, and organic matter concentration increases with sea surface height. But so this was an analysis based on this Hawaii ocean time series, and somehow, partly also to validate this, I'm showing some targeted sampling in environments that can be thought to be the extremes sea surface height conditions. So the lows in the case of mesoscale cyclones and the highs in the case of mesoscale anticyclones. So the anticyclone has a high sea surface height, meaning that the isopical surfaces are depressed in its interior, while the opposite is true for the cyclone. And we had two cruises which organized by, in part by the Simon Foundation in 2016 and 2017, when we sampled in this transect across mesoscale dipoles. They're just showing you some of the a very small fraction of the measurements and observations done during these cruises. And let's start from the nutrients. Do we see the patterns that we had seen in the analysis of the Hawaii Ocean time series? Indeed, we would, not if the graph doesn't show. Anyway, this was one of the cruises. And we, oh, I have another picture. Okay, in 2017, for example, I'm just showing here the vertical profiles of phosphate on the left and dissolved organic nitrogen in the anticyclone center and the cyclone center. And you can see that we see this pattern whereby there is less concentration of phosphate in the surface. And at the same time, we have a lower concentration of dissolved organic nitrogen. So the surface stocks are lower in the cyclone. The same was true in 2016, where we measured just phosphate. And about the vertical gradients, we also measured higher gradients of inorganic nitrogen at the depth of the chlorophyll maximum in the cyclone than in the anticyclone. And so when we look at the phytoplankton community and the abundance of different groups, we can see that between the anticyclone and the cyclone, we don't see much difference in terms of Prochlorococcus abundance. But if you look at the eukaryotic phytoplankton, there is a twofold change in the abundance of these organisms, broadly defined organisms, at the depth of the chlorophyll maximum. And please notice that here, this is not the depth as the distance from the sea surface, but rather I centered this analysis on the depth of the DCM, so I aligned the two ecosystems based on that layer. So somehow these targeted analysis validate the main results of the analysis from the Hawaii Ocean time series. The last point that I want to make here is about this, what we observe in terms of mesoscale variability when we observe it through autonomous observations. And there are several autonomous vehicles that are being used around Station Aloha and north of the Hawaiian Islands, among those Lagrangian profilers and sea gliders. And what they repeatedly have measured is that the DCM of the cyclone is in general, we measure higher concentrations of fluorescence, beam attenuation, and backscatter, so the optical proxies of pigments and particle concentration. And this is not one observation. This, for example, is the median of 600 profiles versus other 600 profiles. And this doesn't always match what we expect from filtration-based measurements. And this is something that we need to solve if we are moving more toward autonomous observations to understand this kind of dynamics. And I think I will wrap it up, but I hope I convinced you that when you see a map like this one of a sea level anomaly, in this case north of Hawaii, you can imagine that there are biogeochemical and ecological changes that are widespread and that are now somehow predictable in part after analysis of this kind. And in this study, some of the main points that I want to make is that we definitely, through the isopical concentration anomalies, have evidence of repeated events of increased new production in conditions of low sea surface height. But in terms of the general changes of the characteristics of a system like Aloha, we know that sea surface height somehow shapes the concentration of eukaryotic phytoplankton likely in association with changes in the diapicnal diffusion of inorganic nitrogen. But also we have these enigmatic results whereby the stocks of nitrogen, dissolved nitrogen and phosphorus change with sea surface height and in mesoscale eddies. And we are still trying to understand what causes this phenomenon. I would be glad to hear your opinion about that. And I think another important point is that we have to solve somehow this mismatch between in situ optical observations and filtration-based observation about the mesoscale variation in the ecosystem status. And I have to thank really a lot of people that contributed analysis, data, and also ideas for, that I presented in this talk. But more than anyone else, Dave Carl and Tara Clemente, and then the funding sources, the Simons Foundation, NSF, the Gordon and Betty Moore Foundation, and the Fondazione Balzan. Thank you.
Time for questions?
So my question is, if you integrate the biomass, not just the chlorophyll, but the actual phytoplankton biomass over the water column, do you see like a big change between sort of whether you're anticyclonic or cyclonic, or is it just sort of like there's a movement in where the DCM is placed, for example?
So in terms of biomass, I think it's rather complicated. I think that you partly said that integrating chlorophyll to get to biomass at sites like stationary law doesn't make much sense because of the big photoacclimation that you have that in most cases is likely to cause the deep chlorophyll maximum itself. From the flow cytometry, there are no estimates routinely done of, for example, forward scatter versus biovolume that allow us to get to the total biomass. So I couldn't do that kind of calculation. We have the stocks of bulk stocks of nitrogen, carbon, and phosphorus in the particular form, and those don't change with sea surface height, apart from the deep layer where we see most likely subduction of those stocks. But if you look at the surface, no, you don't see it.
Ben Twining, Big Old Laboratory. Could you speculate a little more about what might be driving those differences in the optical and filtration measurements?
Well, I think that two main phenomena can drive these changes. I've been reading these papers that say, for example, that eukaryotes, nanoflagellates, are very susceptible to filtration, and they could just not be collected completely, since those are the species that we see peaking at the DCM of the cyclone. If, for example, they broke and the chloroplasts went through the filter, that could cause a mismatch. So there could be a problem of collection of filters. But on the other hand, there's also this thing that the optical efficiency of both fluorescence and scatter is very variable. So if that changed between the cyclonic community and the anticyclonic community, we would see these changes. To me, it's a little strange that we see it both in the scatter and the fluorescence at the same time, because those should be rather independent. But those are the two main hypotheses that I can see.
Philip Boyd, IMAS Australia. Just to come back to this mismatch between the N and P that you showed, that's very striking. If we had an abiotic ocean, it was really just driven by the physics, would you expect there to be decoupled? And therefore, is the biology, particularly in the deep chlorophyll maximum, and some of maybe the differences in ecological stoichiometry, what's causing it?
I'm not sure I understood. Are you talking about the surface stock change?
No, the example you showed for the two extremes of sea surface height, where we see this pronounced decoupling subsurface of nitrogen and phosphorus?
Yeah, let's go back to that. You're talking about this, or for example, this?
Yeah, this one here.
Oh, okay. So at depth, we can agree, I guess, that there is not a big decoupling. And at the surface, you have that while nitrate plus nitrate is depleted, the phosphorus is still at tens of nanomoles concentration, and you have this big change. What I said later, and I probably, this probably wasn't very clear, is that actually also the total dissolved nitrogen stock has the same pattern. There are a smaller amount of measurements of these, including the organic fraction. It's about 10 years versus the 23 years that I'm showing for this. But dissolved organic nitrogen has this same pattern. So it seems that there is a process, and the stoichiometry of the missing fraction of nitrogen and phosphorus is exactly red field. So it seems that there is a missing nitrogen and phosphorus in the cyclonic kind of environment in stoichiometric, in red field stoichiometry.
One more quick question.
Yes, Annie Bourbonnet from UMass London. You know how eddy age would affect those cycles?
No, I don't know about this analysis, and I haven't computed it precisely for the specific cruises. I've been discussing this, and rather recently also with Dennis McGillicuddy, that looking at the eddies that pass through a law and their origin, that their stage in the life cycle could be something that could give us information. And I'm still unsure how much of these changes can be ascribed to nonlinear mesoscale eddies, so those that really trap water. If you look at the maps, for example, if you look at maps like these, well, in this case, a law definitely wasn't in a nonlinear mesoscale eddy. But in several occasions, it happens to be there. It's hard to hit the bullseye. Sometimes it's at the edge. So it could be something important. It could be something that is not driving at least the Hawaii Ocean time series patterns, I guess.